it is the inner layer that provides mechanical strength. So we were looking at the structure, and then we can look at the membrane functions. So we have the membrane, the central layer. It has got hydrophobic. It is hydrophobic, as we saw, and it consists of proteins, cholesterol, and the phospholipids. When we look at the red cell membrane, it is highly classic, can have large membrane extensions without fragmentation. And maybe throughout the work that we have been doing in the laboratory, we have never thought how the red cells is able to maneuver all this without being damaged, because some of them can die due to stress, mechanical stress, but most of them will reach their 120 days. But now this membranes function, their work is mostly or majorly to maintain the red cells, which is the red cells as they do their work. So in the outer membrane layer, they are hydrophilic and they consist of glycolipids, glycoproteins, and the proteins. And in this outer membrane, as we are going to see later, this is where we also find our blood groups anchored, because right now, as we are talking, you might know that there are 47 blood group systems that have been described by the ISBT, and you can imagine you yourself as you are seated there, you are a system because you have all these blood groups. You may not see them, but when we use the identifiers, they can be identified. So they are made of glycoproteins, they are glycolipids, and some of them are carbohydrates. We can call them glycolipids. And then the inner layer completed the one that is next to the hemoglobin. It is hydrophilic and it consists of only protein. As you know that even our heme is a protein molecule. So it is very important to know their work and what they do. They protect the red cell. Others allow a bit of water to come in, and the second layer repels the water, and the third layer is also repelling. So it's very important for the protection of the red cell as it does its daily work. So it's very important for this to be considered when we are considering a red cell membrane function. So what can we say? There are the functions. The main functions that we know is that they carry oxygen from lungs to the cells. The oxygen is carried by a chemical combination with the hemoglobin. As we have seen, there are many pairs, even proteins within this, that they help in the carrying of this oxygen. At the tissue level, oxygen is exchanged with the carbon dioxide, bringing carbon dioxide in the venous blood to the lungs. So the oxygen exchange occurs between 95% saturation in the arteriole with a mean arterial of 95 milligrams of mercury and 70% saturation in venous blood, which means venous tension of oxygen tension of 40 milligrams per. The red cells must be in close contact with the tissue and the successful gaseous exchange to carry, which is done by hemoglobin, the hemoglobin part of it. So it has to maneuver its way next to the tissue so that it can be successful in the gas exchange. Failure to that because of issues that could be associated with it. If the proteins are not working well, if the membrane has been damaged, it may not be able to deliver this. So RBC travels in the microcirculation in 120 days, covering over 300 miles per 480 kilometers per hour. So it's very important to know about this. Just some of these are nice to know, but knowing that within 120 days, senescence will occur, and we'll see how it occurs so that the red cells will be cleared from the circulation so that more new ones can come on board to continue with the activity. And it carries blood group antigens. Actually, the red cell functions also are involved in the carrying the blood group antigens. So how do we understand the red cell integrated through membrane proteins? We know that it maintains osmotic balance and pH support, carbon dioxide exchange in tissues and lungs, And we know that it presents red cell swelling or shrinkage. That is the membrane protein as we had discussed them. And I have tried to show how these roles in the oxygen transport and the hemoglobin role when you talk about the oxygen curve. I know most of you are familiar with the oxygen curve. And then we start with the role of oxygen transport, where it carries oxygen from the lungs, carries oxygen in other body tissues, and delivers carbon dioxide from the tissue to the lungs. And when we come to the hemoglobin role in oxygen transport, it's very important because it's the one that carries this oxygen. We have seen the hemoglobin part of it carrying the oxygen, and it is able to bring 70% saturation. It is able to do the work of removing the carbon dioxide and making sure at 95 saturation, the delivery of oxygen is at its maximum. So it is very important to know about the two of them. They work concurrently. There is none that works alone. And if somebody has got a low level of oxygen, of HB, you know that it will this function will be affected. So it is very important to know about that. But remember, this one is regulated or controlled through the membrane proteins because they have to be there so that they are protecting the red cell from the mechanical damage, and it is able to deliver as it goes along doing its functions. So I was just showing a small picture of the red cell membrane in the capillary. You can see now the figure that we were seeing up there, how it deforms itself so that the biconcave shape can help it to squeeze through the capillaries. And if you look at the first photo where we have got the 4 red cells, you can see the way they are attaching to each other. When they are moving in the capillary, because they don't want to cause any obstruction, they arrange themselves like that so that they are able to move without overlapping each other. So as they go, remember they have got the proteins that are on the surface. As they go through those proteins, they are able to deliver and they are able to pick what they are picking as they move along the capillaries, which are surrounding the tissues and even the veins. So it is very important to imagine in your mind how it does this work as it goes through. So it's very important to see how it can deform itself. But remember that after it has done that, it will go back to its normal shape if these membrane proteins are working well or they are not damaged. So the red cell membrane functions, they maintain shape and deformability. And when we look at MN system, it is a blood group, it is a glycoprotein, and it contributes to the membrane shape. So the protein that we were seeing that contribute to the membrane shape, MNS system is one of the contributor to the membrane shape. We also have the KER system. It links to Macrophage syndrome. And if there is a problem with this KER system, you can imagine we can have some breeding because KER system also is involved in, especially the babies, the newborns. They usually have some breeding on their skin. So the KER system is mostly involved if there is a problem. Then we have the gibbish. It is a structural component. It is a blood group, but it is a structural component of the red cell. Then we have spectrin and crin. BAD 4.5 and 4.2. BAD is the Diego blood group. It is trombolysis the cytoskeleton. And when I was reading about the blood group there before, I never knew the important work that they have in maintaining the red cell shape and even supporting its work as it maneuvers itself through the capillaries. So the function, they arouse red cells to return to shape post-deformation. As it has finished the work, these membranes or the blood groups, they could be blood groups, they help the red cells to go back to its shape after it has been deformed, as we had seen the earlier picture, how it squeezes itself. So I brought this picture so that you can see the blood groups and the red cell. Now, imagine this is your red cell. It is showing you the blood groups that are within, like Lutheran is deep within. If you look at ABO, actually it is on the top because it is a carbohydrate. And you look at Diego, which is also a carbohydrate. So some of them are directly involved past the membrane, and the others, like protein, the RH, you can see they are double within and the outside. Actually, you can see the membrane of the RHs. Some are outside and the others are inside. And this is very important when we are doing our blood groups. You can see Kel. Kel is already a carbohydrate. It is outside completely. You can see YT. It is a carbohydrate. But when you look at gibberish, it is coming directly from the hemoglobin. You can see where it is. As we look at the red cell, the picture we had seen of the red cell, you can be able now to place these blood groups and be able to know whether they are a glycoprotein, whether they are proteins, whether they are carbohydrates, and whether they are both. Because the ones that are extending outside, coming from deep within, remember inside there was protein, and the outside we are having the carbohydrate. So for us, the blood group systems, they are not just blood group to define our blood group. So it is very important. Of course, there are antigens, and there are specific sites on different proteins, glycoproteins or glycolipids. So they are made of proteins, glycolipids or glycoproteins. They form part of the RBC membrane, which the immune system can interact with. That is how now the immune system is able to interact with our red cell. Remember always, the red cell has no nucleus. So, but it does all these functions and it is able to interact with the immune system. So the composition of these blood groups is either carbohydrate, protein, or glycoprotein. And I have given ABO is a carbohydrate, RH is a protein, and MNS, Kel, KID, they are a glycoprotein. So it is very important to know that they play a major role in the maintenance of the red cell as it carries its daily functions. So how do we understand the membrane proteins, such as BAD3 that I said it is the Diego blood group, the CD55, RH-ALAG that controls the RH functions. We have the glycoproteins like the MNS, like KEL, and then we have the DALC like the Duffy blood group. So all these blood groups, some of them are enzymes, some of them are receptors, some of them are anchors, but they are on the surface of the red cell. And actually, these proteins are not only structural, but functionally active. They support exchange, immune evasion, iron transport, and pathogen resistance. And they are found on the surface of the red cell. So blood groups antigen like MNS, KEL, KID, and Duffy are membrane proteins. And we are calling them like glycoproteins, which direct clinical importance in transfusion compatibility and the disease susceptibility. You will see like Duffy is a protective against certain species of malaria. It is not all of them. It is protective against Valley. And I wanted to ask in my own knowledge, when we look at the malaria distribution in our country, we have got like falciparum being almost 93%. When I did the genotyping of Duffy, 93% of the Kenyans do not have the Duffy gene or the Duffy gene. And you can see the percentage of falciparum. You see, Ofale is always given 2% for those people who are familiar with the malaria distribution. So the Kenyan population actually do not have this Duffy gene, and most of them may not suffer so much from the falciparum, and the others are very rare. So it is important to know about this, not calling them blood groups, but calling them the red cell membrane proteins, what they do and their importance. So why does it matter? Knowing how this protein function helps lab scientists and clinicians to interpret blood group types and cross-match results more accurately. understand disease association like PNH, which is associated with the CD55 or malaria susceptibility with Duffy. So there are transport functions in the red cells. The RAG, gas exchange, that is ammonia and the cation exchange. We may never have thought that these membrane proteins on the red cell are involved in this gas exchange. And the band 3, that is Diego, there is bicarbonate exchange, that is chloride shift. We always talk about chloride shift. Know that band 3, that is Diego blood group system, is much concerned about that. AQPI, that is the cotton blood group. It is a water channel, and we looked at the water in the outer membrane, and also about the protection in the inner membrane. KID blood group is known as the urea transporter, and the maintaining of osmotic balance, that is pH regulation and the carbon dioxide transport. That is how these blood groups are very important. You can see there are a lot of iron exchange here. There is water channel, there is urea transport, and then we have got even the cation exchange being performed or executed by this membrane on the red cell. So it is very important to know about this, as even we do our chemistry, as even we do our blood groups, as even we do our hematology, it is very good to know about this unique cell called the red cell. So when we look at the immune defense and the complement regulation, we have chroma, Duffy, or CD55, protects against complement. But if we go back to our, you can see, when we go back to our CD55, there is also the PNH that has the issue of the PNH. And then when we come back here, we have got still the CD55 also found in chroma or DAF, protecting against complement attack. And you know, in blood transfusion, not even in blood transfusion, even in immunology, we usually have complement activation. And this one leads to hemolysis of the red cells. So we have NOX, CRI, which regulates C5, convertase 3, and convertase 5. We have gibberellin and Lutheran. These are adhesion molecule and the immune modulation. So when we look at clinical relevance, like the PNH, there is hemoglobinuria and the inflammation. So it is very important to know their clinical relevance. Like when we have the adhesion molecule, when we have the convertase, and we have seen in PHN, there is that complement activation. And then there will be hemolysis and, of course, inflammation. So when we look at the pathogen entry and the signaling functions, we can see the blood groups or the red cell membranes are very important. Like Duffy, it is a chemokine sink or a receptor, and the malaria receptor. So you can see it is a receptor. Chemokine sink, it is also involved in the chemokines. And most of the times we may never think whether they are involved or not. But I think it is important for people to know this. Lutheran is adhesion and the signal transduction. So it is used in signaling, and also it is an adhesion molecule or anchoring. KEL, it is zinc endopeptiditis, meaning it does the work of cleavage. So zinc is cleaved by this KEL protein. So it is, RBCs also serve as decoys and the signal transducers in immune response. They can express the TLR binding, and activating the innate immune system can also bind and regulate cytokines. So it has got many, many functions, this red cell. It can work as signaling, and actually it can activate the innate immune system. And we know that the ABO have got some functions in the innate immune system because of the existing of the natural antibodies that are within our systems. So let us also think on that line again. So how healthy RBCs avoid phagocytosis? How do they avoid phagocytosis? They have a lifespan of 120 days. They express CD47, which binds CPA on macrophages. So the macrophages have a receptor called CIRPA, which actually is recognized by the CD47. So when the CD47 binds to the macrophages, it sends a signal. They have got their language, which says, don't eat me signal. Actually, the red cell, through the CD47, it binds to the macrophages through the receptor SIRPA, and the communication that is sent there, it is do not eat me. And then it is preserved, and the function of RBCs are preserved. That is how they will avoid to be eaten by the macrophages, especially the spleen, the liver macrophages, or the Kupffer cells. So this signal is very important for them. And the CD47 is very important for them. So when the 120 days are over, of course, the red cell has to be cleared. What happens to this CD47 so that now it does not send the signal of do not eat me? So we enter in the process of senescence, by which the red cells become worn out and leading functions decline, and the eventual removal of these old cells or mechanically destroyed will happen from circulation to maintain hemostasis. Because if there is a lot of red cells bursting, the hemostasis will have an imbalance, as we looked at how it maintains the hemostasis in our earlier slides. So if there is the overstay of these old cells, there will be a problem. So they must be removed. And purpose of senescence in RBC, why does it have to happen? Safe cell turnover. It marks old or damaged RBCs for safe removal by macrophages, preliminarily those ones in the spleen. Also prevents hemolysis, that is, avoids rupture and release of free hemoglobin into the plasma, which can be toxic. And it supports hemostasis by ensuring only function RBC remain in circulation for effective oxygen delivery. So senesced or damaged RBCs, what triggers them? Actually, they lose the CD47, which leads to the exposure of PS or phosphatidylserine, one of the outer membrane layer protein. Then the Diego or the BAD3 clusters. Then there is oxidative damage markers, those ones that prevent oxidative process. They are damaged, the proteins are damaged, and often opsonized with natural antibodies. And now, because we have seen the red cell can signal some activities, now the antibodies will be opsonized, and this one will be targeted for removal. So first of all, there is loss of CD47. There is exposure of PS. There is clustering of BAD3, and the damages that occurs to the oxidative markers. And then the natural antibodies will take over. And then these red cells will be targeted for removal. So there is this membrane changes. I needed this one. PS are a major component of cell membrane, forming the bilipid layer or the lipid bilayer. It acts as barriers to regulate the passages of substances in and out of the cells, plays a role in various biological processes, including signal transduction, forming of specialized lipid structure and the cell structure and functions. The loss of functional proteins, that is senescent RBCs, cause decrease in the activity of enzymes and membrane transporters. Remember, we have the glucose 6 phosphate. That one activity will decrease, and once it decreases, you know our red cell is at risk, and then because it will not have energy, and also the membrane transporters will be affected. They are no longer protected. The PS is the protector layer. Once it is exposed, it will signal the macrophages to come and they eat this, because the CD4 already was lost. The CD47 was lost. Eventually, this leads to impaired oxygen transport and the cellular functions. So the PS is very important. This membrane is the one that causes or changes it together with the loss of CD47. Immediately, the CD47 is lost, even in stored blood, because I'm talking about even the storage of blood where you say 42 days, 21 days. Actually, what happens at that time? The CD47 functionality is lost. And what happens? There is exposure of this PS, and all the others follows now. So maybe we can look in this diagram. We have the macrophage. We have the CILPA that we talked about. It is one of the receptor, and it is the one that is attached to the red cell, because the CD447 density is high and it is not lost. When this one is not lost, the red cell is maintained. But now when you look at the other side, phagocytic clearance of RBC, you can see the CD47 density is low or clustering. So what happens is that once the CD447 is lowered or is low, then the message of do not eat me is not there. So the red cell is exposed to the phagocytic cells or macrophages. And whatever happens, it will have to happen. So there is expression of certain membranes, like CD4 integrin-associated protein. They prevent phagocytosis, but when it is downgraded in senescent cells, makes cells more susceptible to removal by the splenic and the liver macrophages. So it is very important to know how the red cell starts its journey to be eliminated or removed from the system. So in summary, key features of the RBC senescence is morphological changes. Once the proteins are defective or the PS is exposed, there is loss of biconcave shape. Then if we lose the biconcave shape, of course, reduced deformability. The red cell, it cannot go back to its former shape. So they will start forming other shapes like spherocytes, and then there will be increased cell density. The cell will be heavy. It is not able to move, and they will start aggregating together. We saw that they follow each other, but in case there is this, they are not able to do these functions well. Then biochemical markers, there is exposure of PS. And then there is decreased 47. Sorry, there is a lot of noise. There is a lot of noise, sorry. There is decreased CD47, don't eat me signal. increased expression of CD95, the first regard. Actually, this is the death regard, because already there is a call. The CD47 do not eat me signal is no more there. Then caspase 3 activation, modifying the protein that is for death. So this cell will be marked for death. And at this juncture, I don't want to go into details how the iron will be extracted by the macrophages and it is returned to the liver and the rest. But I just wanted to show you the process, how it happens, what is exposed, what happens, so that you can have an idea of what happens. And then after we have all done this, and we have activated the first regard or the death regard, now we go to the erythrophagocytosis. Because it is an RBC, it is erythrocyte phagocytosis. So once these cells are recognized and removed by phagocytes, macrophages of the spleen and the liver, they are removed via this process of erythrophagocytosis. I am not going to read what it says, but it helps to maintain a healthy blood volume and remove damaged and old red cells. Of course, during the mechanical transfers, some of them die. They die before their age, but they have to be removed. So as we are talking, it is not something that even now as you are seated there, this process is happening. Some red old cells are being removed, the new ones are coming into, and even those ones that were not able to complete the functions that they are required due to damage, oxidative stress, they are removed. And then it becomes part of normal RBC turnover and the pathological hemolysis. And then there is central to iron recycling and the immune regulation. So this process, in one way or another, it is very advantageous to our systems and also to the red cell functions. Because if it is central to iron recycling and the immune regulation, we are safe. And the part of normal RBC turnover and the pathological hemolysis, because if there is no this turnover, we could be suffering from pathological hemolysis. So it is very important that it occurs and happens. So the receptors driving phagocytosis, we have TAMS or trial 3, AXL, and MA or MATEC. T cell immunoglobulin and mucin cell surveys. We have even, it binds the PS. FC gamma, we talked about the antibody opsonization, they detect the IgG. So it's very important that we have them. STABI1 and 2 integrin, adhesion and the scavenging, they are liver-based. And we have seen the phagocytosis occurs with the macrophages of the liver and the spleen. So it is very important to know where they are based and the work that they are doing. So occurs in liver, in spleen, liver, bone marrow, and the inflamed tissues. Inflamed tissues, we have talked about a red cell that may not be able to reach its final life or end of its function, and this one needs to be cleared. Maybe it has been stuck in one of the veins or the capillaries and it has to be cleared. So it is very good to know about these receptors that drive this type of phagocytosis. So the macrophages involved in erythrocytosis are of their location. We can have the liver, they are the red pulp macrophages, and their role is to filter aged RBCs. We can say aged and damaged. Do we have the liver, Kupffer cells? They clear opsonization. We can see the antibodies where they are and the damaged red cells. We have bone marrow. This is the central nerve macrophages. They remove defective erythroblasts. And the other tissues, tissue-resident macrophages, these are the ones that keep on walking around. They are like our ascaris. They are tissue residents. They are there in inflammation and the infections. So they are very active in cases of inflammation and infection. So it is very important to know these are some of the macrophages that are very active and involved in the phagocytosis or erythrophagocytosis. I just showed this, which is very important. Loss of self-signals, it may not. Exposure of senescence signals, receptor-mediated binding by macrophages, as we have seen, and gap at the digestion through phagocytome that are found within the macrophages, and the iron released and recycled. So their young red cells are very intact. They have the CD47, and they said the receptor, they attach to the receptor of the macrophages, the one that gives the sign, do not eat me. But the moment it is lost or decreased, then the sign of eat me not is gone. And now the macrophages can do their work. And we are seeing the receptors that help in actualizing this. So it is very important sometimes to look at all the proteins that are involved. You can see we have CD36, we have VCR, CD16, and this is very important, and we have CD51, and I can see STAB2/1 in process, and we had talked about them. This is the process that accomplishes the erythrophagocytosis. It's not just a one step, it is a system, and it goes systematically. So in summary, this erythrophagocytosis is vital for RBC turnover and iron hemostasis. It involves multiple macrophages types and specific receptor ligands interactions, and clinically important in anemia workup, hemolysis, infections, and the human disorder. And we can look at clinical significance. What do I expect if there are red cells, too many of red cell or phagocytosis that is happening? We have hemoglobin being low. Reticulocytes could be early, high, meaning early compensation, meaning there is a lot of asking that we need more cells to be produced because the hemoglobin level is low. And then we have high lactate dehydrogenase, and we will have even high indirect bilirubin. And this is signs of hemolysis. That is when we are doing a full blood count. And then when we look at the peripheral blood film, we will have spherocytes, we will have nucleated RBCs. So sometimes when you see these things, when you are doing a film, if people do blood film today, you can still start thinking about the red cells phagocytosis. So we have macrophages ingestion of RBCs seen in bone marrow films. I would have brought that here, but there is a very big red macrophage that is ingesting small red cells. Then we can talk about Hens bodies because of the oxidative stress. So sometimes you see these bodies, but you may not place them with anything. But it is very important to start thinking about them, so that when you see spherocytes, there is a lot of mechanical damage to the red cells. Nucleated red cells, they represent reticulocytes, and you can know that there is high demand if you start finding them in the peripheral blood film. So it is very important to know. And this peripheral blood film mostly is done from the venous blood, but sometimes when you do a bone marrow, you can also do them smears, and you can stain them, and you can still get these things. So what about disease association? Autoimmune hemolytic anemia by warm or cold and autoantibodies because of the antibody opsonization. So there is clearance of this. There is a new term here, HLH. Actually, this one is caused by overactive macrophages engulfing RBCs and the WBCs at the same time. So overactive immune system that attacks the body's own cells and tissues, that you can say it is one of the autoimmune disease because the body is attacking its own body cells and tissues. So we have infections like malaria, we have sepsis, EBV, then which we have not done much on it, and immune activation or direct damage. So these ones can cause immune activation or direct damage. Like the malaria, you know, it will go to the red cell. Once inside there, it will attack or make the red cell to burst out. So it is very important to know some of the infection that can also be associated here. At the transfusion reaction, there is autoimmunization resulting to hemolysis. There is another disease called storage pool disease. It's a very rare disorder, but it is characterized by defects in platelet granules. So sometimes when you are doing a platelet count or in your cutters, you might get some platelets are more, and then you may not be able to place them. So it is very important to know this one can also occur when we have got the damages of the red cell membrane structures. Of course, chronic inflammation, there is release of inflammatory mediators. And we have seen that the red cell also interacts with the immune system in most of the times. So it is very important also not only to think about the white cells, even the red cells also interact with the immune system. Having said that, because there are receptors on the red cells, it is also important to know, sometimes back when HIV came, people were thinking that if you watch the red cells, you can transfuse them, they don't have the HIV. No, it is wrong, because we have known today they have receptors and they interact with the immune system, and they are the major cells that do most of the work in the body. So it is not true that we cannot have transmission of viruses or other infectious agent through the red cell. It is very important to know that. Relevance in diagnosis and patient management. Link structures and aging of red cells to clinical conditions like autoimmune hemolytic anemia, infections, transfusions reaction, storage pool disease, and the membrane protein defects. And you can remember the PNH, you can see hereditary phagocytosis. We have also some malaria. We also, we can talk about other diseases like cholera. I don't want to bring them there. There are many because of the red cell membrane and the association of its damage or during the time the red cell is being taken out from the system. But if the red cell membrane are intact, even during that process of erythrophagocytosis, not much damage happens. But if they were damaged, then there could be a problem. What about diagnostic clues? Bone marrow aspiration, that is storage macrophages with engulfed RBCs. I do hope that some of you have been able to see that. Flow cytometry, CD163 expression. These are macrophages markers, and you can know whether there is overactivity of them if they are beyond the normal reference numbers. Then we have serology, Coop's test positive in IHA. Then we can do iron studies. Ferritin could be high, serum iron may be low or high. So these are some of the diagnostic clues that can tell you if the erythrocytosis is okay or it is overboard or overreacting, so that you can be able to know if there are problems with some of these membrane proteins that we have talked about. So in clinical management, you treat underlying cause, immunosuppression in higher antimicrobial in infection. Then you can give supportive care, especially in transfusion when indicated, and iron monitoring. So when we talk about hemophagocytic, histolytic, LHL protocols, they should be in place so that they can talk about how they can be managed by the following drugs. It could be etoposide, steroids, and targeted therapy. So it is very important to know about these things when we are doing our full blood count, or when we are doing, sometimes you do some of these lab tests and you don't find the reason why some of these things should be high. But now you can start thinking about the red cell membrane and the work that they do to maintain the shape and the integrity of the red cell. So the takeaway message is erythrophagocytosis is a natural phyto process for red blood cell turnover and iron recycling. However, changes in macrophage activity can alter this process, leading to red cell destruction that signals diseases like hemolysis, infection, or immune disorders. So recognizes these changes through lab tests, through blood count, blood films, Coombs tests, iron studies, and bone marrow exams make erythrophagocytosis a powerful tool for diagnosis and management. I call for action. Be vigilant for signs of altered erythrophagocytosis caused by macrophage stages. Integrate lab findings such as from full blood count, peripheral blood film, Coombs test, and bone marrow exams with clinical insights to detect a disease, and use this understanding to guide timely and targeted treatment improvement for better patient outcomes. Thank you. Back to you, Chair. Hi, Bruce.
Thank you so much, Daktari. I think it's now a QA questions. And I will start with a question from Faye. The question goes, how can we rule out causes of severe chronic anemia in a patient with normal RBC count? Over to you, Daktari.
How can we rule out causes?
How can we rule out causes of severe chronic anemia in a patient with normal RBC count?
Okay, thank you. The patient has normal RBC count. Now, I wanted to ask, have they ruled out the other issues like infection, like cancer that is affecting the white cells, so that they can be able to know if the bone marrow is working? They can do a bone marrow aspirate. They can know what are the cells or what is affecting the shutdown of what is bringing this. And then most of the times we never check on the iron levels. So I don't know whether they have checked about the iron stores, and then to know whether there are other underlying causes that could be bringing this type of anemia. Because sometimes you may have a red cell count that is higher defined. People may have some other issues relating to oncology and the other things, because somebody with the oncology, and even when people had the first cases of the HIV, actually, they were coming to see that people were having this when the HB could be a bit okay, but as the disease progressed, that is the time now the HB was going high. Other causes could be the cause of having this chronic anemia, but I am suspecting that there could be an internal hemolytic process that could be going on but the bone marrow is working well. And somebody have to do even a blood filling to establish how these red cells are, even if we find them normal. And at this point in time, I would like to answer, we first of all also look at other causes that could have brought that. Back to you, Bruce.
Thank you. Fred, are you able to speak? Okay, okay. Anyone with a question, maybe you can raise your hand. Okay. Thank you so much, Daktari. It seems no more questions. Or someone is asking, someone is asking,
yes,
what is the relationship between HIV and low HB and red cell count? What is the relation?
I just want to say, because of the immunosuppression or compromised status, the production of all cells, once there is immunosuppression, the bone marrow could be suppressed as well, and it is not able to produce enough cells of all types, actually. In cases of immunocompromised diseases, HIV that is not being treated, we can have cancer cases, we can have other cases that actually destroy the red cells. But when we talk about HIV, we are not going to talk about only the red cells alone. But remember, when the immune system has got an issue, it may affect most of the organs and the functions. I have seen somebody asking for the notes. I will share with the MLS care so that they can be able to give the notes. How can you describe false anemia in relation to spots, and how can they be managed? Actually, spots consume a lot of energy, and red cells are also stressed sometimes when you subject them to a lot of mechanical work. And number two, it also depends at which level we are describing. If there are higher levels and the lower levels, you will get different types of HB. And how can we manage the anemia in relation to spots? First of all, you have to manage yourself so that you are well. And then there is this about minding what you eat in relation to the false anemia that we are talking about. So I think I have tried to look at the Q&A answer, and I don't see any other. Maybe you can tell me if there is one I have left. Thank you. Back.
Yes, another question. What are the factors that can lead to production of antinuclear antibodies in humans? That's a question from Kelvin Owino. Over to you, Daktari. What are the factors that can lead to production of antinuclear antibodies in humans? You can unmute.
Okay, I got you. The factors that can lead to antinuclear antibodies in the body, in the antibodies in human.
Yes.
I think when we talk about antinuclear, we have to know what they lead to, the antinuclear. And the moment we know what leads to the antinuclear, we are able to know, is it the lifestyle? Is it an underlying condition? Because some of them, because they bring about inflammation and the other things, it is very important to know. As even we want to dive deep into this, we need also to know where we are, the location, the lifestyle, and even how we seek, what do we say, healthy seeking behaviors. It's very important. You have to, I can't say that, I can say it is poison. You see, the lifestyle, there are people who are using chemicals and the others. There is this issue of a lifestyle that maybe doesn't have some exercise, and you start developing other autoimmune diseases. So there are many factors that can lead, but we have to check on the lifestyle. We have to check on the health seeking behaviors, and we have to check on our location where we are. If you are in a location where there is exposure of toxic substances, it could also be contributing to that. And for now, Bruce, that is a good question that need also to be looked into further. But I can tell you, we may not say for sure this is the factor that really leads to the nuclear formation of the antibodies. Thank you. Back to you.
Maybe the last question, because there are several. The last question from, this is an anonymous attendee. What is the relationship between glucose 6-phosphate dehydrogenase deficiency and malaria?
Thank you for that, because this enzyme is the one that gives the red cell the energy or the strength, actually, because the red cell do not have a nucleus which is supposed to be commanding all the processes of making sure that the red cell has got some power or energy. So what I can say for now, it is inside. We found that it is in the deeper part of the red cell. And the red cell, actually, the malaria goes directly into the red cell. I can say, because some enzymes work as receptors, could be it initiates the reception of the malaria parasite into the red cell. But I'm not saying for sure that I have looked into that in depth. Thank you, and back to you.
Thank you so much, Daktari. I think we have come to the end of this session. It was a great honor, and we thank you for your time. And to the participants, thank you so much for the time. I know and I believe we will improve with your input, with your contribution. Our emails, our communication channels are open. You can still forward in your contributions on how to improve on these. Otherwise, thank you so much. Your certificates will be issued within 72 hours. That is the normal TAT for us. Issuance will start immediately. So just wait.
Thank you so much. Thank you so much, Bruce.
Someone also requested that we bring on board an officer from KMLTB to explain to us on how to self-report. Yes, we will do that. We'll strive and do that in our next session. Otherwise, thank you. Daktari, you can have your closing remarks, then we end it from there.
Thank you very much, everyone, for listening, and thank you for the good questions. And you know that the red cell, once it is destroyed by the malaria, actually there will be hemolysis. And when there is hemolysis, the platelets are called upon. So don't forget that the hemolysis that occurs, whether it is internal or external, it is injurious. And so the platelets will be called upon by the mechanisms of the protection or of the immune system. I'm happy and honored to be your presenter this afternoon, and I'm hoping that at least we have learned something about the red cell membrane and how the red cell is able to be maintained, and even as it maneuvers itself through the thin or small capillaries, considering its diameter. So always, if you have a question, actually, you can ask them through this forum, or you can reach out to me and we can have more discussion on this. Otherwise, thank you very much for this opportunity. I'm also looking forward to discuss with you more about other topics that are of great importance to us. Thank you very much.
Thank you.